Hello, everyone. Good morning. It's my pleasure to introduce Mark Velleca from G1, and he will tell us about myelopreservation and multilineage.
Good morning. Thanks, Gil, for that introduction. I appreciate the opportunity to share the G1 story with everyone here in New York City today, and also like to welcome those joining via webcast. So our forward-looking statements. Our vision is to improve the lives of those affected by cancer with 3 investigational therapies, each of which addresses a distinct unmet medical need. We've stayed focused on doing what's right for patients while operating in a capital-efficient manner. And as we move towards commercialization, we expect to form partnerships to make our therapies available to patients globally. So our pipeline has 3 investigational therapies. First, we'll cover trilaciclib, which is a first-in-class myelopreservation therapy. Last year, we reported positive data in multiple phase 2 trials, which we'll review today, showing that we can reduce myelosuppression and improve safety for patients on chemotherapy. This approach is different than lineage-specific growth factors and transfusions. We are in the midst of regulatory discussions and expect to report the outcome of those regulatory discussions and the pathways to approval for trilaciclib in the second quarter of this year. Our second product candidate is lerociclib, a differentiated oral CDK4/6 inhibitor for which we presented proof of concept data at ASCO last year. I'll review those data quickly. Key differentiating features of that drug: less dose-limiting neutropenia and the potential for less CBC monitoring. Favorable tolerability profile makes it, we think, an ideal combination agent in indications outside of breast cancer, such as non-small cell lung cancer, for which we have a trial ongoing with Tagrisso. G1T48 is our potential best-in-class oral SERD. This drug is differentiated chemistry, which we think is going to mean better tolerability and safety. It's an order of magnitude more potent than Faslodex. It's currently in a dose escalation trial in patients with ER-positive breast cancer, and we expect to report the first clinical data, proof of concept data, on this investigational therapy in the fourth quarter. Obviously, having an oral SERD gives one a significant opportunity to benefit patients across multiple lines of therapy. Current status of the pipeline: the 4 phase 2 trials for trilaciclib, all of which read out last year, 3 in small cell, 1 in triple-negative breast cancer. The 2 trials for lerociclib, the oral 4/6 inhibitor, currently in dose expansion phase 2, in combination with Faslodex, and dose escalation in non-small cell with Tagrisso. And the SERD, G1T48, monotherapy in second, third line patients with ER-positive breast cancer. So let's turn to trilaciclib. And what's pictured on this cell are hematopoietic stem and progenitor cells. So these cells are the reservoir from which all blood components are formed, so red cells and white cells. And these cells get damaged by chemotherapy, causing anemia and neutropenia. So patients are at risk for infection, they're fatigued, they require growth factors to stimulate the production of neutrophils. Those growth factors cause bone pain, and transfusions to replace the red cells that the anemia. So trilaciclib protects these HSPCs and therefore preserves all cells, white cells and red cells. The current state for patients undergoing chemotherapy is they get myelosuppression and require rescue therapy. So there are high rates of neutropenia and anemia starting at 12 o'clock on this slide, requiring red blood cells and ESA rescue, hospitalizations, chemotherapy dose delays and reductions, transfusions not just of red cells but also platelets, neutropenia that can cause febrile neutropenia requiring G-CSF, and so multiple interventions to support and rescue these single lineages. Trilaciclib is given up front before chemotherapy, preserves these HSPCs, reduces, as I'll show you in 3 randomized studies, anemia and neutropenia, and reduces those interventions, the transfusions and the G-CSF usage. So we think this is a better option for patients. It addresses a substantial unmet need. 1 million patients in the U.S. still get chemotherapy. It's not going away. It's being used increasingly with IO regimens, and myelosuppression is still prevalent. We're going to review very shortly the phase 2 data that shows less neutropenia and anemia, reduced G-CSF usage and transfusions, and a PFS benefit in triple-negative breast cancer because those patients were able to receive more cycles of chemotherapy. Next steps, as I mentioned, is a regulatory update that we'll give in the second quarter of this year, and we're already exploring partnerships to maximize the opportunity for this therapy globally. These are the 3 trials in small cell lung cancer. The first was in a first-line setting with what is the standard of care, carboplatin etoposide. All 3 of these studies were randomized, placebo-controlled, double-blind, and they were global studies conducted both in the U.S. and Europe. The second trial here, also in a first-line setting with carboplatin etoposide. The new standard of care now is with Tecentriq. This has now been approved. So we were already in that setting, a 107-patient trial. And then on the bottom row here, a second, third-line study enrolling patients with topotecan plus trilaciclib or topotecan plus placebo. So this slide has a lot of data. We'll take our time and walk through what it shows. First, there's 3 histograms that are in bundles across the slide. The orange histograms are the first-line carboplatin etoposide data. The dark blue is the carboplatin etoposide Tecentriq, and the Carolina blue is the second, third line. These histograms represent the decrease of bad things that happen to patients when they get chemotherapy. So the deeper these histograms go, that means the more reduction of these bad things happening on chemo. And those are all listed at the top of the slide. Duration of grade 4 neutropenia in cycle 1, a number of different neutropenia endpoints including G-CSF, and then some of the red cell endpoints towards the right of the slide. So what you can see is the consistency across all 3 studies. You can see that many of these were met with a high degree of statistical significance. And very importantly, these are clinically relevant endpoints that have regulatory precedence. So let's just start with the very first bundle of histograms. Mean duration of grade 4 neutropenia in cycle 1. This was the regulatory endpoint that led to the approval of Neulasta. Every day that a patient has a neutrophil count below 500, i.e. grade 4 neutropenia, they have a 10% risk of getting febrile neutropenia. So if you can reduce that duration, you reduce the risk of febrile neutropenia. And we did indeed see that in this study in the 5th column down. Patients had access to G-CSF in both arms of the study. We followed NCCN and ASCO guidelines. And you can see that the patients on the trilaciclib arm had fewer G-CSF administrations per cycle. Even with the G-CSF that was administered to the placebo patients, they had more febrile neutropenia. So a better option for neutrophils. Importantly, because of trial's mechanism, it helps more than just neutrophils. Also, a red cell benefit was seen, whether you're looking at anemia or red blood cell transfusions, and what's boxed here is transfusions after 5 weeks on therapy. That was the regulatory endpoint for the approval of the ESAs. Overall reduction in grade 3-4 heme AEs, and importantly, reduction in dose reductions, chemo dose reductions. It's important for a therapy like this not to impair the efficacy of chemotherapy, and that was clearly shown in all three studies. And the most important measure of this is a hazard ratio, do no harm. We've seen response rates, PFS and OS, that are consistent between the two arms, comparable between the two arms, and consistent with the historical literature. But it's those hazard ratios that are well below 1 that clearly demonstrate that we are not impairing the efficacy of chemotherapy. There are some settings where we may actually improve the efficacy of chemotherapy, and this is indeed what we saw in the triple-negative breast cancer study. So this was another randomized study where patients received gemcitabine carboplatin. These are metastatic triple-negative breast cancer patients in either the first, second, or third-line setting. And what we noticed is that patients getting trilaciclib received significantly more cycles of chemotherapy than those in the control arm. The safety profile was consistent with the small cell trials, and there were no trial-related SAEs. The PFS, the Kaplan-Meiers are shown here. You can see that the hazard ratios are about 0.5 for both of the trila arms. When you combine those trila arms, you reach near statistical significance for an extension of PFS of about 2.5 months. So what's next for trila? As I mentioned, we're in the midst of regulatory discussions. We expect to give that update on what's next, what's the pathway to approval in small cell. This is a drug that can be used across multiple chemotypes and tumor types. The next two big categories of chemotherapy, taxanes and fluoropyrimidines, we expect to address in label expansion trials. We are also following the potential for trila to benefit survival and looking at the triple-negative data, expect to report mature data later this year, as well as the ability for trila to potentiate the effect of checkpoint inhibitors with the Tecentriq study. Also expect to report data later this year. From a commercial standpoint, what are the key takeaways? Again, still substantial unmet need. There have not been any significant innovations, and chemo remains the backbone. Based on market research that I'll quickly review in a moment, multi-lineage myelopreservation in small cell is a substantial opportunity, $500 million to $1 billion worldwide peak. Physicians like the proactive approach. In fact, in the market research, those physicians that were the highest G-CSF prescribers were the ones that really liked the profile. They wanted to use this up front, and they saw it as a better approach for patients. And payers clearly put it in a different category than growth factors because it's not just addressing a single lineage. It addresses multiple lineages. And they saw cost savings based on reductions in hospitalizations, transfusions, and growth factor administrations. This opportunity to expand beyond small cell makes this a potential multi-billion-dollar peak product. So most importantly on this slide, patients are better served with proactive myelopreservation. G-CSF causes bone pain. Red blood cell transfusions take a lot of time. Patients get allosensitized. They are not transfused typically in this day and age until their hemoglobin is below 8 and they feel lousy. So they will do better, we believe, on trilaciclib. Physicians see trila as different than what they currently have, and payers see the multi-lineage benefits based on a budget impact model that we shared with them. So that all builds to this large commercial opportunity. So let's switch gears. I'm going to talk about our oral 4/6 inhibitor, lerociclib, and then I want to finish with the SERD. So what makes lerociclib different in the CDK4/6 landscape? It's primarily PK. We can dose this drug continuously. Ibrance, the market leader, requires a 7-day holiday, as does Kisqali. We can dose this drug, we think, with less dose-limiting neutropenia, and what we're currently exploring is the potential for patients to have less CBC monitoring for neutropenia, which would be a clear differentiating factor versus Ibrance. The drug is safe based on the phase 1b data we presented at ASCO in combination with fulvestrant in ER-positive breast cancer patients. We don't have the QT liability that Kisqali has, or the drug-induced liver injury liability of Kisqali and Verzenio. So no monitoring for EKGs, no LFT monitoring, much better diarrhea profile than Verzenio, and no propensity for VTE. So favorable tolerability and safety and the potential for less monitoring are what makes this unique. The data we presented at ASCO last year will be updated. We'll have the full escalation data set in the fourth quarter of this year. We also expect to have our randomized phase 2 dose identified and again exploring this potential for less monitoring. Just to quickly review the data that we shared again at ASCO. On the left part of this slide are the neutrophil curves. So you can see at the various doses the neutrophils decline and then they plateau. And this is with continuous dosing. So we're not changing anything once patients fall below 1,000 ANC, which is grade 3 neutropenia, unlike the 3 marketed drugs, all of which have either stepped up monitoring or dose reductions or dose delays on grade 3. We dose through that and we see this plateau. If you look at the very bottom dotted line on this slide, that's grade 4 neutropenia, and we see less of that. So you do see a decline. That's a pharmacodynamic effect, but we can dose through it. We've seen anti-tumor activity at all dose levels. We know it's an active drug. It's more potent than palbociclib. And the preliminary response rate data is on par with the first data readouts from PALOMA-3. So these will all be updated with over 36 patients worth of data. There were only 24 at this point last year. So what other opportunities do we have for lerociclib? It could be combined with other targeted therapies, and that's exactly what we're doing with Tagrisso. Opportunities in prostate with an AR antagonist. There is actually some clinical data with the BTK inhibitor ibrutinib and mantle cell. So multiple opportunities here that we expect to be able to explore in partnership. The current status of the Tagrisso study, it's in dose escalation. We expect to present the early dose escalation data in the third quarter of this year, that you can combine these two drugs without overlapping toxicity. I want to finish, let's finish with the G1T48, the oral SERD. And we'll have much more to say about this drug over the course of the year. We think this is a potentially best-in-class oral SERD. It's more potent than Faslodex by an order of magnitude. It has differentiated chemistry that we think will give favorable tolerability. We expect to report proof of concept data in patients with ER-positive breast cancer in the fourth quarter, and dose escalation is ongoing in that trial. We have a very rapid development plan in place. All of the required non-clinical and CMC studies are either underway or planned. We expect to be able to identify the randomized phase 2 dose at the end of this year and be in a position to begin randomized trials in the first half of next year in first line, second, and third line. And this is an enormous opportunity for patients, translating to a big opportunity commercially, both as monotherapy in second, third line setting and in combination with CDK4/6 inhibitors in a first line setting. So patients can benefit across multiple lines of therapy. So another dense slide. This is actually from Hope Rugo's review on the ASCO guidelines a couple of years ago. And the point of this slide is that starting even in the adjuvant setting, there's an opportunity for patients to benefit from an oral SERD. All they have now are aromatase inhibitors, tamoxifen, fulvestrant, which is the only SERD on the market, but it's intramuscular. It's a very painful set of injections. Patients describe extreme pain to the point where they can't sit down. So having an oral SERD that's better tolerated, that's more potent, that has better exposure, we think can drive better efficacy and certainly better tolerability. And that can translate to patient benefit in multiple lines of therapy, not just in the second, third line setting, but in a first line setting and potentially, and this makes it an even larger opportunity, the adjuvant setting. So our development plan for G1T48 so that we can get a standard of care label. By the end of this year, we'll have presented proof of concept data and expect to be able to determine the randomized phase 2 dose. We'll take a parallel strategy, monotherapy in a second, third line setting, and combination with CDK4/6 inhibitors in a first line setting. Again, potential to benefit patients across multiple lines of therapy. The current trial is in phase 1 dose escalation. Primary endpoints are essentially safety and getting that randomized recommended phase 2 dose. So we'll have that by the end of the year. Data that you expect to see would be PK, PD, and that's assessed by FES-PET. Preliminary response rate data. We're also doing a food effect to look at bioavailability. The milestone timing, again, escalation ongoing, and anticipate sharing those data and the recommended phase 2 dose in the fourth quarter. So key takeaways for these two oral drugs. Lerociclib differentiated from the other three 4/6 inhibitors. Potential for less monitoring, and we've already seen less dose-limiting neutropenia. Favorable tolerability profile, we think will make it a partner of choice in combo regimens beyond breast cancer, and expect to see more data on this drug in the fourth quarter. G1T48, our potential best-in-class SERD, oral delivery providing a significant advantage to patients versus Faslodex. Opportunities both monotherapy and in combination with 4/6 inhibitors, and you'll see a proof of concept data for that in the fourth quarter. And finally, combining various aspects of our pipeline, 48 and 4/6 inhibitors, an all-oral breast cancer regimen that patients don't have right now, a SERD plus a 4/6 inhibitor. We believe that this will result in better outcomes. We've got those studies designed to look at randomized activity of an oral SERD versus standard of care in both the first line setting, second and third line. As far as milestones across the pipeline for the rest of the year outlined on this slide, we will be presenting additional data on 3 of the trials for trilaciclib. A second, third line study in small cell for which we press released data in December. We'll present additional data this quarter. We'll provide that regulatory update, as I mentioned, on what the next steps are for trilaciclib development and ultimate approval in small cell. And then the other 2 data points are driven, the other 2 presentations will be driven by data. So pending mature OS for the Tecentriq study and mature PFS for the TNBC study. For lerociclib, initial Tagrisso escalation data in the third quarter and more escalation data for the Faslodex study in breast cancer in the fourth quarter. And finally, the SERD, G1T48, proof of concept data in the fourth quarter. So I'll finish there. Thank you all for your attention. Glad to take questions and look forward to speaking with all of you during the course of the year during this very important set of milestones through 2019.
All right, any questions from the room?
Just, you had mentioned at some point that there was another drug that didn't hit on ORR necessarily, even for butterfly on OS, did actually better. So through, I can go on this,
Yeah, so all of our discussions with FDA have been with the heme division about the reduction of myelosuppression, not about enhancement of survival. And the expectation is if you are a protective drug and are trying to reduce side effects, what they want to see is that you're not impairing the efficacy of chemotherapy. And the most sensitive measure of that is the hazard ratio. So clearly in all of these studies, based on looking at those hazard ratios, we are not impairing efficacy. The response rates in small cell for 30 years, there's been no correlation with response rate and overall survival.
Then what are the different potential outcomes of the regulatory meetings that keep coming up? They change cycles, wanting more data. What are you asking for?
Yeah, well, we're talking to them about what's next for trilaciclib development to ultimately get an approval for small cell, and the indication is reduction in myelosuppression. So
that's just about size of the trial, or what are the key questions that get hashed out?
yeah, well, we don't share the key questions that we're going to be asking regulators, but we'll certainly share the outcome of that meeting. I think it's the usual kind of questions that you have after you've done 3 randomized phase 2 studies about what's next to get the drug to patients as soon as possible.
But again, in a registration trial, you don't expect that they'll be asking the primary.
I can't speculate. We haven't had the meeting.
Is immune cell suppression a major issue in upcoming IO therapy? Is this like something doctors have seen?
Yeah, I think it's interesting. No one's paid attention to lymphocytes, the fourth lineage. There's clearly a literature on chemotherapy-induced lymphopenia that can be long-standing. I think the other immunosuppressive part of chemo is red blood cell transfusions. Red blood cell transfusions clearly are immunosuppressive. So we just had an ad board a few weeks ago in New York, and one of the docs was talking about how the way we give chemo now in general is immunosuppressive, and given that chemo is being used with checkpoints, Trila could be a nice adjunct to that regimen. So that's indeed why we did the Tecentriq study, and we'll see what the outcome of that is later this year.
And maybe the last one. Have you guys seen results showing that less neutropenia, less dangerous neutropenia corresponds to, let's say, the hospital?
Well, it would in the sense that there's clear data that there's less febrile neutropenia, and a febrile neutropenia episode typically gets you admitted to the hospital. And in fact, in some of the trials, we did look at hospitalizations, and there was a reduction in all-cause hospitalizations, not just due to febrile neutropenia. Yeah.
Thank you. Any other questions from the audience?
Great. Thank you all.